Hi everyone, it's Mr. Vallejo. Today we're going to take a look at mitosis. As I share the screen with you, let me remind you that all of these PowerPoint slides are available for you in your learning management system, whether that is Canvas or it is Schoology. So let's go ahead and get started on today's lesson. So mitosis occurs in just about all of your 38 trillion cells. And what mitosis is division of the nucleus, but it's really division of the genetic material inside your nucleus. So as we study mitosis, what we know about mitosis is partially influenced by what we know about how it happens in bacteria. So we're going to take a look at cell division in bacteria, and then we're going to apply the lessons that we learned from prokaryotes to eukaryotic cells, and we'll see that cell division is a little more complicated because we have a larger genome, and we'll take a look at the chromosomes, which have a different structure than the chromosome that is in the bacterium. We'll take a look at the term diploid and haploid before we go and take a look at the cell cycle as a whole, and then we're going to take a look at just that little part of the cell cycle where the chromosomes are dispersed to different nuclei. That's mitosis, and we're going to take a look at the different stages of mitosis. After the nucleus divides, then the cytoplasm is also divided, and so that is called cytokinesis, and we're going to take a look at that as well. So cell division in bacteria, you should know that the bacteria, just like you do, have DNA, and that DNA is double-stranded. You can see in the picture right here, here's a circular DNA that's not in a nucleus. We also have a smaller circular piece of DNA, which is optional. It's called a plasmid. Some bacterium have a plasmid. Another bacterium might not have a plasmid. That is a lot smaller than this primary bacterial chromosome. We'll see that in eukaryotes, the chromosomes are not circular, but they're more X-shaped. Now, replication occurs or begins at the origin, right here. This is the letter O for origin. Sometimes in other diagrams, you'll see it as RO for replication origin. And from the replication origin, there is bidirectional synthesis. That is to say, DNA synthesis occurs in both directions, that the copying of the DNA starts here and then it goes both to the left and to the right in this diagram. When it first starts out, it looks kind of like this in an ideal situation. And so because this looked like to someone the Greek letter theta, we call this also theta replication. In bacteria, it is size that is a key factor, which tells the bacteria that it is time to split. So when it splits into two, we call that binary fission. Binary means two. Fission means to split. This is a type of asexual reproduction. And asexual reproduction, remember, if it's something is A, it's without. So this is without sperm and egg. So it's asexual reproduction. Now, eukaryotes are a little bit different. Eukaryotes are larger, and eukaryotes have many more genes than bacteria do. So they're much more complicated. Instead of just having one round chromosome, a eukaryotic cell is going to have multiple chromosomes. And those multiple chromosomes are all located in this control center of the cell, in the organelle called the nucleus. The chromosome structure has well, let's take a look at the chromosome structure. And most of the time, you don't even see the chromosome. In this case, this is when we're just starting to see the chromosome. This is at the first stage of mitosis. But it says individual chromosomes contain a very long DNA molecule associated with proteins. We're going to see in the next few slides that the DNA is wrapped around these proteins. But it's not really thick. It's not what we call condensed. And so we don't see the individual DNA. We don't see the individual chromosome at this point, but it looks fairly uniform, the nucleus does. And what we do see is what we call chromatin. Chromatin ends in IN, so it must be a protein. It's the chromosome form of the, it is the DNA wrapped around these proteins, and this is the form that you'll see the DNA, the genetic material in for most of the cell cycle. You won't see it as chromosomes until these guys are just ready to split that chromosome and deliver the parts to different nuclei. Now, as far as chromosome structure goes, the chromosome structure depends on the species. You and I have 23 pairs of chromosomes. That means we have 46 chromosomes in each of our 38 trillion cells, with the exception of those few sex cells, those sperm and those eggs. We're going to take a look at that when we study a similar process called meiosis. So in mitosis, the chromosome structure that we're going to see in different species is consistent in individuals. Individuals of the same species have the same chromosome number, obviously. So you and I have 46 chromosomes. Lily plant has 12 chromosomes, 12 pairs of chromosomes. But this number, just because it gets higher and higher, doesn't mean that it has to do with complexity of the organism. Look at this one, 39. That's a chicken. A chicken has 39 pairs of chromosomes. So are you saying that a chicken is more complicated than I am? Well, I'm not saying that at all. I've had a pet chicken. We had a pet chicken named Brownie, and it would always forget it could fly. We would let it go in the backyard, and my Labrador retrievers would run in circles trying to chase and get this bird, and the bird would keep running in circles. And all he had to do is fly up to the top of the fence, and he would have been done with it all. One time, that same bird, I was eating a piece of pizza in the backyard, and that bird landed right on top of my head, started pecking at my pizza. Stupid bird. That's a chicken for you. Chickens, not that bright. 39 chromosomes, though. Are they more complicated than 23 chromosomes for humans? I don't think so. Chromosome structure, the number is consistent within the species, but it does not have to do necessarily with the complexity of the species. Here's a diagram which shows you what happens after that chromatin starts to condense. That chromosome is going to start getting thicker to us. And see, here's the DNA. It's wound up, but then it gets wrapped around these histones, and the unit is called a nucleosome. So now we have these nucleosomes that are going to produce a fiber, and that fiber gets wrapped around some more. Let's look at the next slide. It tells us more about that, because here's the fiber. This is called a 30 nanometer fiber, and then we take that smaller fiber and we wind that up some more. It's almost like you had a rubber band, and then you ever play with a rubber band and do that and make it as small as you can? Well, it's a little bit like that, because they say, and there are different ways to illustrate this, but if you were to take one of your DNAs and unwind it and stretch it, it would go for miles and miles and miles from here to San Francisco and back. So very far distance. So that just to illustrate the magnitude of concentration of the DNA. You have this long string, and gets wound up and then wound up and then wound up, and it comes all together, and finally you get all this information into the chromosome that we see at the very bottom right corner. Now, there's a lot of information. Humans are made up of 30,000 genes, they think. So Uh, that seems like a lot of information to package into 46 chromosomes, 23 pairs of chromosomes, really? Well, it's turned out that a lot of the space is actually not used for genes. We call that space the heterochromatin. So there's two different pictures, two different um, diagrams that show us DNA. So in this one at the bottom here, called heterochromatin, the black line represents DNA, and it surrounds the protein, which is the yellow disc right there. And then here over here in this diagram, this is euchromatin. It's the EU, which remember is good or true. It's true DNA. It's the DNA that actually says something. So here's an enzyme called DNA polymerase, and DNA polymerase is reading this, and then that is part of the process of creating the protein product that results from reading the DNA. So the euchromatin is a good chromatin. The heterochromatin is a different chromatin. It doesn't have anything to say. It doesn't have any genes. It's almost like it's the Interstate 5, the freeway. If I'm taking the Interstate 5 from Southern California up towards Sacramento, I would see a lot of emptiness, starting from where the I-5 and the 99 split. A few miles after that, if you need gas, you can go to Buttonwillow, and then the next exit, there's a bathroom. But then after that, you've got to drive like 45 minutes to get to anything else. You can stop at Lost Hills. But if you take a look at the side as you go in between those stops on the freeway, seems like there's the same mountains. Seems like there's the same cow. Seems like the same power line going, but just, oh, it's endless. But every once in a while, there's a stop. DNA's like that. Every once in a while, there's a stop. And that stop might be Coalinga. And if there's some place that's significant on the chromosome, and it means something. So that's the euchromatin. Those are the genes. So the chromosomes look like an X. I said in comparison to the bacteria chromosome, it looks like an O. But this whole thing is a chromosome, but it's actually two copies of the chromosome. Those two copies are called chromatids. So here's one chromatid, and here's another chromatid, and these two chromatids are held together by the centromere. This diagram is a little bit different from the one before, in that it shows the gene also. The gene is a part of the DNA which actually codes for a protein product, so it codes for a trait. It wouldn't necessarily have a purple band there or anything like that, but sometimes we can add chemicals to the DNA, and then bands form. Sometimes the genes will be right on the band, sometimes the genes will be off the band, but you can use that band to say, well, this gene is very close to this band, if not on it. The process of karyotyping is important to chromosomes. And what we see here is this is an old photograph of a karyotype. But what it shows us is that you have different chromosomes, and they are in pairs. And there's 23 pairs altogether. There's 22, and here's the 23rd pair. And they're grouped according to size. We'll learn later that the 23rd pair is a little bit different. It's called the sex chromosome. And this is the karyotype of a male. The male is XY, we say. Yeah, female is XX typically. And this karyotype we might do from a fetus. And so it says here it uses fetal skin cells from the amniotic fluid surrounding the baby. Just like you and I lose a lot of skin cells. What are the two examples that come to mind are I've heard that 40 to 50% of all dust is of human origin. And I've also heard that if you take your mattress is after 7 years weighs almost twice as much as what it used to weigh. because all the skin cells that we lose every night. Isn't that interesting? Well, the baby then is also losing skin cells, and we could take advantage of that. And because the skin cells have nowhere to go except the amniotic fluid, you can take that amniotic fluid and then run the karyotype to figure out if the baby has enough, the correct number of chromosomes. So does the baby have 46 chromosomes? Is it missing a chromosome? Does it have an extra of something else? We can learn a lot by doing a karyotype. So here's a couple of diagrams that'll help you understand what's happening. Here's a sonogram, and here's the fluid being withdrawn from the mother. Of course, you don't want to hit the baby. They have other ways to do this now. They can do chorionic villus sampling, or CVS, which is less intrusive and a much quicker test. And that is what you do with the karyotype. We'll see more on that a little bit later in the next lecture on meiosis, but you also have a lab exercise to complete that has to do with karyotypes. Before we get to the cell cycle, let's take a look at haploid and diploid. The term haploid means a cell that has only one chromosome of each pair. So this theoretical organism has 3 pairs of chromosomes, but in this cell, you only have one copy of those cells, or one copy of those chromosomes. In the diploid cell, you would have 2 copies of each of the 3 chromosomes. So you have 6 chromosomes total, and you have 3 pairs. We have 46 chromosomes altogether, and we have 23 pairs. So in humans, we have, in a haploid cell, we have 23 unpaired chromosomes. A silly way to remember this is diploid, di means 2, so diploid cell has twice as many chromosomes as a haploid cell, and a haploid cell has half as many as a diploid cell. So haploid and diploid. Now, the cell cycle consists of 3 major phases here. We have interphase, which is the longest phase of the cell cycle. Then we have mitosis, and we have cytokinesis. So we're going to take a look at each of those, but here's the major points of interphase, mitosis, and cytokinesis. You can see that during interphase, we're going to duplicate the chromosomes and then make some cell parts to get ready to split. Then mitosis, the chromosomes that were already duplicated are going to be distributed to 2 different daughter nuclei. After the nucleus is divided, cytokinesis occurs, and that's when you take the cytoplasm and split the cytoplasm in half. Cytokinesis not as exact as mitosis is. So it's helpful to remember the stages of the cell cycle. So here's a mnemonic that I give you. I put my arm there. I put my arm there tells you that there's interphase, prophase, metaphase, anaphase, and telophase. Some of you might be thinking, well, what about cytokinesis? Well, then let's add Cynthia to here. I put my arm there, Cynthia. That's interphase, prophase, metaphase, anaphase, telophase. This part right here is mitosis. Some people remember mitosis as PMAP, so that works for you, great. So remember, the whole cell cycle is not just made up of mitosis, but the whole cell cycle is interphase, mitosis, PMAP, and cytokinesis. So that's why we have, I put my arm there, Cynthia. Now let's take a look at the interphase in particular. Interphase is the time between mitoses. It's most of the cell cycle. It's been likened to, if the cell cycle is 24 hours, then interphase is 23 of those 24 hours. It's the time between mitoses, and to further our study, we further subdivide the stage of interphase into different phases. And so we have G1, G stands for growth. Before they knew more about it, they they thought that the G stood for gap, but now we say that it's for growth. So during G1, you have general cell growth that occurs right after the cell forms from the binary fission. Then we have the S phase. The S phase is, the S stands for synthesis. Synthesis of what? Synthesis of the DNA. The DNA is going to be synthesized or made, and so you're going to double the amount of DNA in the S phase. You don't even see them as chromosomes yet. This happens while the DNA exists in the form of chromatin. In G2, we're getting close to getting ready to divide. So the growth stage, which is different from G1, that the cell is getting ready, it's preparing to divide. So because this takes a little more energy, we see that G2 is a little bit different from G1 because in G2 you have replication of the mitochondria specifically. So that's interphase. During mitosis, what we're going to see is this duplicated chromosome, which has two chromatids, is going to split. So that's what's going to happen. And then these chromosomes are going to go to different nuclei. And then finally, in cytokinesis, after we split the nucleus, we're going to split the cytoplasm. More on that in a little bit, but that's cytokinesis. Now, here are the four stages of mitosis: prophase, metaphase, anaphase, telophase. Remember, that's PMAT. You should know these characteristics of the four different stages of mitosis. This is a picture of prophase. During prophase, there are three things you need to remember. First thing is that the nuclear membrane disappears. The second thing is that the chromosomes are going to start to condense. And thirdly, you see these structures right here. These are the centrioles. The centrioles start out right here, and they're going to start moving towards the opposite sides of the cell. We say they're opposite poles. So those are the three things that happen during prophase. The chromosomes condense, the centrioles move to opposite poles, the nuclear membrane starts to disappear. Then what happens is the chromosomes start to line up in the middle. We call that the equator. And so we call this meta or middle phase. It's a very short phase. Out of all of these phases, this is the shortest phase. But it's significant because you can see they're starting to line up. The next phase is anaphase. During anaphase, what happens is the chromatids, now called the chromosomes, move apart. The moment you see a gap between the chromatids, they are now chromosomes. And those chromosomes are going to move towards opposite poles. Silly story, I remember this from when I was a freshman sitting where you are. The way we remembered anaphase was I was in the marching band and we had 250 people in the marching band. I played saxophone. I was in the back row because our band director was a trumpet player. So he always put all the brass instruments in the front, but all the woodwinds went in the back. And you know, when you're in the marching band, you got 249 other friends. Well, like at 248, because there was this one flute player, her name was Anna, and she just was always in a foul mood. And no one wanted to be next to her. So you wanted to move apart from Anna. So that's how I remember that when I was a freshman. When I was sitting where you are, I remember that mitosis, anaphase, Anna move apart from Anna. So that's what happens during anaphase. And telophase is the opposite of prophase. So the three things that you need to remember for prophase, remember the opposite for telophase. So the nuclear membrane is starting to disappear, the chromosomes are starting to disappear, starting to not, the opposite of condensing, they're disappearing. And then the centrioles are moving back so that they're not at the poles any longer. Another thing you start to see this little bump where the cells are starting to divide, and we call that cleavage. We'll see that come into play completely as we look at cytokinesis. Now, this is an animal cell that's starting to go through cytokinesis, and see that what's called the cleavage furrow right here. The cleavage furrow occurs when you have the cytoskeleton component called the microfilaments, and those are starting to pull those it's starting to get tighter between the cells, and it cuts it off. And so that's what you see in this diagram at the bottom right here. So in animals, cytokinesis occurs by constriction of the cell called cleavage, cell cleavage. And that doesn't occur in plant cells because plant cells, you have a cell wall. So you can't have that, the pulling of the string that you see in animal cells. So instead what happens is you have this cell plate starting to form right there. Once it becomes complete, and you've got your two cells. And so you have cytokinesis. Now, practically speaking, why is it useful to know about mitosis as far as medically speaking, and in the laboratory? You can see here that most normal cells divide only when attached to a surface, and they keep going until they start to touch another cell. So right here you can see the cells are growing and growing, but finally they stop because they're touching each other. Well, we found out that the addition of a protein called the growth factor can influence these guys to keep growing. And that's what cancer cells do. Cancer cells don't know when to stop. So does this have something to do, the growth factors, does this have something to do with cancer? Well, if you take a look at this diagram here, you have checkpoints, and there are biochemical checkpoints, and you have to have a certain protein in order to get past this point. So in a way, we can stop the cell cycle, and we can stop the cell cycle, we can stop cancer cells from continuing to grow. And so that is a very good thing that gives us hope. So cancer cells are just cells that forgot how to stop. And there's something going on with their exterior proteins on their cell membranes, on their glycolipids and their glycoproteins. They aren't working effectively or correctly. And so they don't know when to stop. And when you get the tumor continues to grow and grow and grow, and then finally it's nowhere else to go, so it breaks off and goes into other tissue. When that happens, we say that your cancer has metastasized. And so when it metastasizes and goes into your bloodstream or to your lymphatic system, that's not good. And so hopefully we can go and take our information about growth factors and cells being proximity of other cells and use that for a cure. Because I would love it if we could cure cancer in our lifetimes, don't you? All right, so I'm Mr. Vallejo, and that's Mitosis, and I hope you learned something today. We'll see you next time. Take care now. Bye-bye.